I'm Tanya from Dr. Omics. So, in today's session, I'm going to tell you about the enrichment analysis, which is basically divided into two parts: pathway enrichment analysis and the functional enrichment analysis. So, what is the pathway enrichment analysis? It means in which pathway the gene is highly enriched. So, that we are going to find out here. So to find out this information, we are going to use one of the database, that is the KEG mapper, color and search pathway. So in this tool, here you can provide the gene names, KOS or the EC numbers if you want to retrieve any information regarding the pathway. So for that, you have to open the your data, right? So in the previous session, we already find out the annotation of our data. So just open that txt file. So this is the file which you have generated from the Proca tool. So in this tsv file, you can see there is a locus tag they have provided, feature type, length in the base pair. This is the gene name and the EC number. So with this EC number, we are going to retrieve the pathway information. So what we can do, simply just copy this EC number and paste in another sheet. Then just select this A column, go to data, then there is option sort. Now simply just copy the entire EC number and paste that in this box. Now execute this. So you can see total 109 pathways are involved, and below that pathway name they have given along with their pathways IDs, and in this bracket how many number of genes are involved, that you can see the number here. Now if you want to store the information regarding the pathway information, so what you can do, just simply copy the entire data and paste that in Excel. Now select this A column, again go to data, there is the option of sorting. Now what we can do, we can separate the number of genes and the pathway name, right? So just select the A column, type Control H, and you can replace this with hashtag. Now just again select the A column, type Control H, or maybe what you can do, just select the A column, data, text to column, delimited. Next, in the other you can put hashtag, then finish. So you can see it has separated the number of genes and the pathway names, and so you can see citrate cycle, like the other name of the citrate cycle is the TCA cycle. So you can just delete this, just simply copy this number and delete. Now again just select the B column, Control H, replace this, and insert one row, and you can write here name of the pathways, and here you write number of genes involved. Done. Now for example, if I want to work on any of the pathways, So you can see here is a list of the pathways, right? Along with the number of genes involved. So for example, I want to work on this ethyl benzene degradation. So we can open with this ID. Now you can see the EC number which you have provided, it is highlighted in this color, in this red color. Again, if you want to like download this figure, you can just download this image in the PNG format. And if you want to get more information regarding the CC number, you can just open this. You can see the symbol name, the entry ID, and the name of this, along with the gene names that are involved in the pathways. And if you want to cross check with your data, that also you can do. This is your EC number, right? Just go to this main file, just select the C column, Control H, and search this number. So you can see this is our EC number, and here the gene name is the FADA, right? So if you want to cross check your information whether it is right or wrong, you can see FADA. Can you see? Along with that, here you can see the COG ID, or you can see the COG link they have provided. So this is the COG ID that also you can see here, that is the COG 0183. So now for example, if you want to just work on this data, you can just simply insert a figure. Yeah. So this is how we do the pathway enrichment analysis. We are going to see the function enrichment analysis. So basically this is divided into three parts: molecular, biological, cellular. So in this we are going to see our gene is having the which functions, right? Or how many number of genes are involved in which functions. So that we are going to see with the help of one database, that is the Shiny GU. So here in the Shiny GU, we have to provide the gene list. So in the Proca tool, you can see just simply copy the gene name and paste that in this box. Submit this. Now if you want to get the only the biological process, you can update this. So this is your biological process. Along with them, they are providing you the FDR score also, and the number of genes data from our list. And if you want to download this data, you can just select on this result on this pathway. And again, if you want to update on the cellular component, it will be update to the cellular. If you want the molecular function, so this is your molecular function. Now for example, again, if you want to like make any plots, any pie chart, maybe bar plot, so for that you have to open this file which you have downloaded from the Shiny GU. Just open that. in the Excel, delimited, next, comma, finish. So you can see entire information is there in this Excel now. This is the FDR score, the number of genes and the pathway genes, fold enrichment score again. We are not interested with this. So we are only interested with the pathway information and the number of genes involved. So what you can do, just simply copy the number of genes and the pathway information. Now you can sort this data, go to data, then sort on the basis of number of genes, largest to smallest, make it okay. Now, for example, I want to create a plot maybe for this top 25, because you can see the like number of genes that are more in this. So again, it's up to you, like for which you want to make a plot. You can also add more. So for example, I want to make the plot for this top 25. So just go to the insert option, and there is a option of chart, pie chart, or any other bar plots also. You can see that bar, right? Maybe area. So it's up to you which type of plot you want to create, you can create. So for example, I want to work on this bar plot. Just select that, make it okay. Now you can see the number of genes that are highlighted with the different colors here, right? So if you want to like present the number of genes on the chart, what you can do, just right click it, go to add data labels, then add data labels. So you can see here the number of genes that are involved is the 11, right? And you can see it is highlighted, which is respond to the external stimulus. So likewise, you can also create the plot for the biological also, molecular also. Yeah, thank you.